Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about another case study, unusual source of contamination during SDS-PAGE. And here's the problem. I followed the exact protocol for kidney, liver samples and it's worked. But it's not working for brain samples. I extracted the histone proteins using pre-lysis, lysis and balance buffer from Bio-Rad. I'm not going to read the rest, it's just description of the protocol, which is not important for our today's discussion. So take a look. Here is this gel electrophoresis. So this is polyacrylamide gel electrophoresis. And as you see, there is a lot of contaminants here. But what is the source of the contamination? Let me underline that for kidney and liver samples, this technique worked. But it didn't work for brain samples. So what's the difference between the tissues of these two organs and this organ? Actually, probably it's not occur to many of you, but brain contains a lot of lipids, which other tissues also have, but in much smaller quantities. And also some of you may think that lipids are non-polar, so they shouldn't move at all and shouldn't make such artifacts what we see here. But actually what may happen here, that proteins which were loaded here, actually when they moved, they just left this, I would say, trail. Just like, for example, people, if we step in something oily and would work, we also would leave a trail of footsteps, oily footsteps. So this is exactly the same what we see here. So that's why this is unusual source of the contamination. And also I want you to take a look at this picture of another gel electrophoresis and take a look. In the well number 1 and 2, we also have here protein extract taken from the oil seed. And here's the ladder, and what you see in the lane 3 and 4 is the same samples which we loaded in wells 1 and 2, but this time after lipid removal. As you see, resolution here jumped, I would say, like tenfold. It is much more clear bands than what we see here. So what methods we can use in order to get rid of this lipid contamination? The first one can be organic solvent extraction. Yet another variant can be detergent-based extraction. Third method can be precipitation method. And you also have to perform dialysis or desalting after, if you choose any of this method. After removing lipids, it is important to exchange the buffer of your protein sample to suitable buffer for SDS-PAGE. I will provide a brief description of each method in the video description, so you can also check video description, but protocols you probably can find online. I'm not going to provide links for those protocols because all these protocols always refining and you can choose the best protocol for your own situation. And this is all for today. Subscribe and see you in the next video. Goodbye.